I'm back with the another session on photosynthesis, and in today's session it will be very important for you all. It will be full of concepts because today we are going to learn about the Z scheme, and this Z scheme is very complicated, as you all know. Every time I used to get the students like, said, how can we remember this, how can we remember that? But don't worry, today each and every concepts, each and everything will be clear to you, and I promise you that you're going to have zero doubts after this session on the Z scheme. Okay, so फटाफट से अपना अपना सारे लोग अटेंडेंस लगा दो. Give me your attendance. How many of you are present? Hi Shivesh, good to see you. Hi Shri Sai, good to see you also. Great. Start kare. Should we start? Okay. How many of you are comfortable with Hindi and English? Yes? Should I ask you some questions? Shri Shai, from where you are? Shivesh, from where you are? Okay, great. Are you typing something? Actually, comments comes little bit late to me. Okay, you are from Delhi. Great. Okay, chalo. Let's start this session. Let us start this session. So before that, let's revise whatever the things which we have discussed in yesterday class, because yesterday class was kind of, very interruptive. So we'll discuss the Manipur. That's great, Sri Shai. Okay, I love the northeast place, but I haven't visited there. I'm that kind of person who loves to travel. I hope that I'll come to Manipur very soon, right? Okay, chalo, let's quickly get started. So we discussed that in photosynthesis, the photosynthesis takes place where? The photosynthesis takes place in the, what is going on? Yeah, something happened, na? I remembered, yeah. How is the condition now? Is it fine now? See, so photosynthesis is what? Photosynthesis takes place in chloroplast. Yes, sir, we discussed it yesterday. In the chloroplast, sir, we have a special structure which were known as thylakoids. Yes, so I'll quickly revise all the things. Let's suppose this is the chloroplast. Okay, let's suppose this is the chloroplast. Inside the chloroplast, we have what? We have small structures which are known as granum. Okay, all this was known as what? All this was known as granum, and the single thing is known as what? The single thing is known as the thylakoid. Yes or no? The single thing is known as thylakoid. Just a second, guys. The single thing is known as thylakoid, and the stack is known as what? And the stack is known as granum. Yes or no? Clear, sir? Okay. Now what we did is that we took out one thylakoid. And we saw that the cell membrane or the membrane is what? It is made up of lipid bilayer. Yes or no? The membrane was made up of lipid bilayer like this. Remember, we discussed this thing. I'll show you the diagram. 1 second. Yes. See, we saw that the membrane of thylakoid is made up of like this, the lipid bilayer. Yes or no? In this, we had photosystem 2 and photosystem 1. Inside the lipid bilayer of thylakoid, we had this photosystem 2 and photosystem 1. Which one was discovered first? The PS1 was discovered first. But in working, which is working first? So PS2 is working first. Clear? These things we have discussed yesterday. Okay. Now, from here, let's start it again. So, in photosystem 2, what we have seen was that there is the chlorophyll A in both photosystem 2 and 1. But what was the major difference here? The major difference was that this photosystem 2, the chlorophyll of photosystem 2 says that I'll absorb the light of 680 nanometer. And the chlorophyll of photosystem 1 says that I'll absorb the light of 700 nanometer. Then, sir, we have a confusion here. What about the other lights? What about the lights having 400 nanometer? What about the lights having 500 nanometer? So those lights were absorbed by these accessory pigments. What were these accessory pigments? The xanthophylls, chlorophyll B, carotenoids, all these pigments were the accessory pigments. They were absorbing the other lights which were not absorbed by the chlorophyll A. Okay, and all the lights were absorbed, and Light was exciting the electrons, so all the electrons were given to this particular reaction center. Clear? This much we discussed in the last class. Sri Shai, any doubts still here? This much we discussed in the last class, right? Okay. Now, what happened is that this is what? This is the chloroplast membrane. Okay. This is the chloroplast membrane. And inside the chloroplast, we had this thylakoid. So, inside the thylakoid, we have a space, and this space is known as what? This space is known as lumen. Clear? Okay, sir. This space is known as lumen. Now, this space is what? This space is known as stroma. Okay, great, sir. This much is clear. Where is the photosystem present? Photosystem present here. Photosystem 2, oh my god. Let me take this one. Photosystem 2 and photosystem 1. Okay. I hope that we have revised all the things which we have discussed in the last class. Okay, great. Now, let's move on and let's talk about this thing. This was the thing, right? जहाँ पे हमारे साथ scam हो गया था last class में. My green screen got started its tantrum and internet was started his tantrum, right? So let's start it again. So what I'm doing is that I'm trying to make a chloroplast. Okay, this is a chloroplast, right? And one second. Okay, now whatever you are doing, stop everything, focus here. Your focus should be here, right? This is the chloroplast. Okay, and this is the thylakoid. Now what I am doing is that I am just making the thylakoid and erasing the chloroplast. Okay, I am, see very carefully, I am erasing the chloroplast and I am making the thylakoid big. So this is what? This is a thylakoid. This is what, sir? This is the thylakoid. Clear? Okay, great. Now what? In the thylakoid, we have what? We have photosystems. So here, let's suppose that we have photosystem 2. Let's suppose we have photosystem 2. Okay. There is one more photosystem, which is what? There is one more photosystem, which is what? PS1. So PS1 is present here. Let us make it with the help of this red color. This is PS1. Okay. Okay, sir. This is so clear. Now what, sir? Now what? Now we are going to have the magic. Just imagine that light is coming. Okay. Just imagine that light is coming. Light will bring what? Light will bring energy. And this energy is going to excite an electron from the pigment which is present in the photosystem 2. So what was the main pigment present in the photosystem 2? It was chlorophyll A. Yes or no? Main pigment of photosystem 2 is what? Chlorophyll A. Clear to everyone? Now what happened here is that as soon as the light came here, the electrons of chlorophyll A got excited. As soon as the light came, the electrons of chlorophyll A got excited. Okay. Electrons of chlorophyll A got excited. Clear? Electron came here. Okay. Excited, came outside. Like sometimes we say, our mother scolds us that, okay, don't do this thing, don't do that thing. You just sit and watch reels and all of these things. And then you will be like, okay, I'm going out from my home and I'm not gonna come back. But when you go out and then you realize that you don't have anything, right? Then you have to come back to your home. So just like this electron now it came outside and now it's thinking that now where to go. So, but this electron found a home which was primary electron acceptor. The name for this is pheophytene. So very firstly, after coming out from the chlorophyll A, where it has gone to pheophytene. Okay, pheophytene is what? Primary electron acceptor. Pheophytene is the first person who has accepted the electron from chlorophyll A. Okay, so primary electron acceptor. right? So what is the primary electron acceptor, sir? Let's write the name. So first primary electron acceptor is at any point of time. If you guys will have any doubts, let me know, okay? Let me know at any points of time you have doubts, any doubts, let me know. So primary electron acceptor is what? Primary electron acceptor here is the pheophysine. Okay, sir, great. Try to remember everything. Pheo, P-H-E-O, pheophysine. Clear, sir? Now what, sir? Now the thing is that pheophysine was like, I don't want to keep this electron for a long time. Pheophysine is going to send the electron where? Pheophysine is going to send the electron to the another complex, which is PQ. And what is PQ, sir? PQ is plastoquinone. What is PQ? PQ is plastoquinone. Let me write it little beautifully. Na? Here what we have? We have pheophysine, okay? We have pheophysine. First of all, pheophysine. Okay. Now this pheophysine gave the electron to whom? Gave the electron to PQ. And this PQ was also like, no, I don't need the electron. I'll pass to the other person. It passed the electron to cytochrome B6 complex. It passed the electron to cytochrome B6 complex. Okay. Okay, sir. Now what? Now the cytochrome B6 was also like, I don't need the electron. I'll also pass to someone else. And it came to PC, which is plastocyanine. Sir, how can we remember this? We are small, cute little kids. How can we remember so much of the things? Guys, so you have to remember, you don't have any other option, right? So how you're going to remember? See, just remember, chlorophyll A, energy came, electron got excited, it went to the pheophysine. From the pheophysine, it went to the PQ, PQ to cytochrome B6, cytochrome B6 to PC. Clear? Now, you can remember it like this also. Queen comes first, okay? So, PQ come first and PC, which is C for Sena, like let's suppose. So, C is coming after the queen, okay? So, PQ first, PQ is coming, then after that PC is coming. And between them, there is cytochrome. So, this way you can learn. Chlorophyll A, electron got excited. The very first person to accept the electron is pheophysine. So that's why we are calling it as what? Calling it as the primary electron acceptor. Primary electron acceptor. Sri Shai, any doubts? Let me know. Okay. Primary electron acceptor. Why? Because the very first time it is accepting the electron. After this, it went to PQ. After PQ, it went to cytochrome B6 and then to PC. Okay, sir. Great. Now, this much is clear to everyone. This much is clear to everyone. Any doubts still here? Let me know in the comment section. Any doubts anyway, let me know in the comment section, guys. No doubts? Okay. Okay. Let's move on. Let's move on. Okay. Now, you might be having one question. You might be having one question that, sir, this particular chlorophyll A is giving the electrons. Okay. Yes, it is giving the electrons. When the light is coming. From where this chlorophyll A is getting the electrons? Like continuously, it is releasing the electron. So there might be a source of electron, right? Exactly. So the source of electron here is the water. The source of electron here is the water. Yes, pani, right? Okay. So what is happening? This portion was what? This portion is the lumen, yes or no? The lumen of thylakoid, inside the lumen. So inside the lumen, what is happening? Water is breaking down. Inside the lumen, what is happening? Water is breaking down, which is H2 breaks into 2H positive plus O. I need to balance it. Okay. So 2H positive and here we have O negative and we will be having plus 2 electron. Okay. Plus 2 electron. Exactly, photolysis of water. Plus 2 electron. Okay. Now this 2 electron is going here. Now see, this 2 electron is going here. So chlorophyll A is basically doing nothing. Chlorophyll A is getting the electron from the photolysis of water and it is just sending those electrons. Clear? Any doubts? Great. See, we will clear the things, very basic things, and step by step, and we will be very 100% sure, confident in this Z scheme. Okay. So what happened? First of all, we saw chlorophyll A, excited electron went to pheophycine, pheophycine to plastoquinone, plastoquinone to cytochrome B6, cytochrome B6 to PC. Now we had a question, sir, from where the electrons are coming. So we saw the electrons are coming from this thing, okay, from the photolysis of water. Where it is happening? It is happening inside the lumen, right? Great. Now, sir, what about this? After PC, what about this? Like electron came here, then where it is gone? Now, this PC was also like, I don't need the electron, guys, take it from me. Take it, the electron from me. So, after PC, the electron is what? The electron is given to this photosystem 1. After this, the electron is given to the, which color should I use? Red? Yeah. After this, the electron is given to this photosystem 1. Now, I am going to ask you a question here. A very quick question, okay? Which of the following is involved in photolysis, sorry, which of the following is involved in photolysis of water? Let me check who is going to answer this question. Number 1 is what? Number 1 we have is PS1. Second we have is PS2. Third we have both, and fourth we have none. What happened to my handwriting? Fourth we have none. Okay, answer me. PS2. Great, very good. PS2 is the correct answer, because in photosystem 1 is not getting the electron from hydrogen from the photolysis of water, right? Photosystem 1 is getting the electron from the PC, which is plastoquinone. That's very good. That's very good, Sri Shai. Very good. Yes. Okay, great. I'm very happy. Now what? Now this photosystem 1 is again going to get the light, okay, and when the light is received by the PS1, it is also going to excite the electron. Now this electron is again will be received by a primary electron acceptor, which is FRS, okay, which is FRS. From FRS it will go to FD, okay, ferredoxin. Now what is going to happen, sir? Now what is going to happen about the electrons? See, at this point of time, like right now you might be thinking that, sir, how the ATP is made? Till now we are just transferring the electrons from here to here to here to here. At the end I'll say you how the ATP is made, but right now there is one more product which can be made. Just see it nicely. So what I am doing is that here FD is kind of here, present here, okay. So when the electron is reaching here in the membranes of the thylakoid, so there is an enzyme present, which is what? Which is NADP synthetase. What happened? 1 second. Yes. This 6:30 timing is something bad or what? My screen gets lag here. The internet gets something bad. Okay. See. Now we have what? Here we have an enzyme. Outer membrane pe we have an enzyme which is known as NADP synthetase. Which is what? NADP synthetase. Right? Okay, so what does this enzyme do? It catches NADP It catches NADP from the stroma of the chloroplast. It catches NADP from the stroma of the chloroplast, and with the help of this electron which is being released by FD now, with the help of this electron which is released by the FD, it makes what? It makes NADPH. NADPH. One second people, one second. My screen, just 1 second. Okay, yes. So by catching this electron, the NADP synthetase is converting NADP into NADPH. Sir, H kahan se aaya? From where the H came? So the H is used, the hydrogen ions which were present in the stroma, that hydrogen ion is used. So till now what we have discussed, again let's quickly revise, okay, so that there should not be any confusion. So light came, so this PS2, the reaction center of PS2, which is the chlorophyll A, the electron of this chlorophyll A got excited and it went to pheophycine, okay. Pheophycine is what? Pheophycine is the primary electron acceptor, okay, great sir. Now after this primary electron acceptor, it went to PQ to cytochrome B6, cytochrome B6 to PC, great, okay, clear to everyone? Any doubts anywhere? Let me know in the comment section. Now after this PC, the electron was transferred to PS1. In PS1, the electron got excited and primary acceptor in PS1 is FRS, that is this particular thing is accepting the electron, and then the electron is transferred to the FD, okay. Now what happens that the electron reached here, okay, the electron, all this thing is happening where? All this thing is happening between this layer only, within this layer. So the electron reached here. As soon as the electron reached here, there is an enzyme which is called NADP synthetase, and this enzyme is going to catch NADP which is present in the stroma of the chloroplast, which is outside the thylakoid, and with the help of the electron and the hydrogen present in the stroma of the chloroplast, they are making NADPH. Clear? They are making NADPH. Any doubts? Srishai, Shivesh, any doubts? Any doubts till here? Let me know. थोड़ा सा भी doubt है तो let me know. No? No doubts? No doubts, people? Great. Great. If you don't have any doubts, let us move forward. Okay. This much is clear? Yes, sir. Now we also saw that the electron which we are getting, how the chlorophyll A is getting so much of the electrons with the help of the photolysis of water, which was happening inside the lumen of the thylakoid, right? Inside the lumen of the thylakoid. Great. Clear, sir. Clear. This much is very much clear. Okay. Now let us see that how the energy will be made. The main question is that we have to make ATP, sir, and right now we haven't made any ATP. So see, ATP will be made with the help of this hydrogen ions. ATP will be made with the help of these hydrogen ions. How, sir? See, we are going to create a potential difference. We are going to create a potential difference here where between the concentration of the hydrogen ions outside and inside. Just imagine that there is a balloon, okay? Just imagine that there is a balloon and inside the balloon there is lots and lots of air, okay? Obviously, inside the balloon the air pressure is very high, and when you pick a pin and pinch it, the air will move very fast, okay? So in the similar manner, what we are trying to do is that we are trying to accumulate as many as hydrogen ions possible in this particular lumen, okay? We are trying to accumulate as many as hydrogen ions inside the lumen. So there is two conditions. One, so you produce hydrogen ion inside the lumen, right? Second condition is what? You reduce the hydrogen concentration outside. See, when we are forming the NADPH, hydrogen ions is getting reduced. Okay, there can be two possibilities, right? If you want to study, you study. Like if you want to top, you study. Or what you can do is that you can ask your friend or you can disturb your friends so that they can't study, so you will get top automatically, right? So there are the two scenarios. One thing, you produce the hydrogen ions inside the lumen, or You reduce the number of hydrogen ions outside the lumen. The both thing is being done. Hydrogen ion is produced with the help of the photolysis of water, and hydrogen ions is reduced when NADP is produced. Sorry, NADPH is produced. Clear to everyone? Any doubts? Now what? Again, there is third thing also happening. There is one more thing happening which is reducing the formation, which is reducing the concentration of hydrogen ion outside. This PQ is what? They are very clever. This PQ is very clever. It says that when I'll transfer the electron, when I will transfer the electron to cytochrome B6, I'll take one hydrogen from the stroma and will give it inside the lumen. Okay. So while transferring the hydrogen, what is happening? This PQ is transferring one hydrogen as well. From where? From stroma to lumen. From stroma to lumen. Clear? Any doubts? Any doubts, anyone? From stroma to lumen. Okay. So three things are here which is creating the hydrogen potential barrier. Okay. Hydrogen potential barrier. So what? Number one is the photolysis of water. Yes, sir. Number one is the, what happened to my screen? See, my iPad throw tantrums every time. So number one thing is what? Number one thing is the photolysis of water. Photolysis of water. Yes. Photolysis of water is happening where? Let me know the answers in the comment section. Photolysis of water is happening where? It's happening in the, yes, exactly, lumen of thylakoid. Lumen of thylakoid. Okay. And this is increasing the concentration of hydrogen. Second thing is what? Second thing is formation of NADPH. NADPH कहाँ हो रहा है? Where it is happening? It is happening in the stroma of chloroplast. Yes or no? Stroma of chloroplast, which is outside the lumen. Stroma of chloroplast. And third thing, what is happening? Third thing is that this plastoquinone is what? It is transferring the hydrogen from the stroma to the lumen. PQ transferring the, Shri Sai, any doubts? Do you have any doubts? Shri Sai, any doubts? Plastoquinone transferring electrons to the lumen. Right? So can you see that? What is the conclusion here? The conclusion is all the things here. Suppose this is the lumen and this is the stroma. All the things is trying to build a potential barrier and trying to increase the concentration of hydrogen ion inside the lumen and trying to decrease the concentration of hydrogen ion outside the lumen or in the stroma, right? So we have successfully created what? We have successfully created the potential difference between the stroma and the lumen. Clear to everyone? Yes or no? Any doubts? Any doubts, people? Any doubts? No doubts? Great. Okay. Now what? Now something is going to happen. What is going to happen? See, I'm going to make another diagram for it. It will take little time. In this session, we'll just clear out the Z scheme, okay? So that you can go and revise it from the NCERT. I'm not going to give you more concepts. Just the Z scheme, we will clear it out so that this thing will be cleared and you will solve some of the questions. You will read the NCERT and then we'll start the cyclic and non-cyclic thing. Then we'll start the Kelvin cycle and all of this thing, okay? Okay, let's come back here. Till then, the hydrogen ion is inside the lumen. Now this hydrogen ions, when we are going to release this hydrogen ion, the energy will be created. Just like, let's suppose that during the flood, what happens? There is a dam, okay? There is a dam and during the flood that dam gets overflow, and we open the barrier, we open the gate of the dam. So in a very rush the water comes, right? So that water can be that water is having lots and lots of energy in it. So if suppose I put something there, so we can generate lots of energy. So in a similar manner, what is there? There is one protein which is known as F1 F0 complex. Okay, there is a protein which is F1 F0 complex. So this protein is kind of like this here. Where it is present, sir? It is present in the membrane of the thylakoid. Okay, how should I make? Let's see, I am making it with the green color. Okay, this way. There is a protein which is present here, and this protein is known as where it has gone? F0 F1 complex. Clear? Now, through this, the hydrogen is gonna release, and as soon as the hydrogen is gonna release, there is an enzyme present here. There is an enzyme present here which is ATP synthetase. Yes. That is ATP synthetase. ATP, yes, Shrishai. ATP synthetase, okay? ATP synthetase. And this ATP synthetase is going to produce ATP. This ATP synthetase is going to produce ATP. Got it? So this was what? This was the main purpose of this whole Z scheme, to produce ATP. How the ATP is produced? In the stroma we have ADP, adenosine diphosphate, plus we have what? We have the inorganic phosphate. 1 second, I'll decrease the thickness of my pen so that I can accommodate some more things. Is it visible? Yeah. See, we had what? We had ADP plus inorganic phosphate, and with the help of the energy which is generated from that release of hydrogen, they are getting converted into ATP. Clear? See, any doubts? Any doubts still here? So why it is known as Z scheme? Why it is known as Z scheme? See, tung, tung, tung. This is the Z scheme. The shape here was the pathway here is what? This, and this. This is why we call it as a Z scheme. This is why we call it as a Z scheme. Clear to everyone? Any doubts anywhere? Let me know in the comment section. Shri Shai, Shivesh, any doubts anywhere? Any doubts anywhere? Take a screenshot of this, okay? So beautiful diagram, right? Okay. Now, should I make you a flowchart for this? Should I make a flowchart for this so that you can, when you are studying this thing, it should be in your mind, right? See, so first of all, what happened? Chlorophyll A, okay? Chlorophyll A of which system? Photosystem 1 or photosystem 2? Chlorophyll A of photosystem 2. Who said photosystem 1? Huh? Photosystem 1 will work afterwards. Photosystem 2 will work first, okay? Photosystem 1, oh, I am only saying photosystem 1. Photosystem 2, okay? Light came, tuck, 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 tuck. This light came here. The electron got excited. The electron got excited. 1 second. Yes, the electron got excited. Now this electron went where, sir? This electron went to the, where it is gone? Same. Ferredoxin, phycocyanin, PC, where it has gone? Yes, it is gone to primary electron acceptor. Primary electron acceptor, which is what? Which is at this point, it is pheophycin. Yes, exactly. Pheophycin. Check the spelling of pheophycin, okay? Pheophycin. I need to take some English classes to improve my spellings. Yes or no? Huh? No, it's not like that. 1 second. So we have this pheophycin. Clear? After this pheophycin, where it is gone? It is gone to PQ, Rani, Queen. And between PQ and Sena, Rani and Sena, what was it? Cytochrome. Rani और Sena के बीच में क्या था? Cytochrome B6 complex. Okay. And here we have PC. After PC, where it is gone? after PC it is gone to, okay, where should I stand? Yes, after PC it's gone to photo system 1. Okay, now photo system 1 से कहाँ गया? Photo system 1 से कहाँ गया? Tell me. Yes, photo system 1 से कहाँ गया? Tell me in the comment section fast. सारा कुछ मैं ही बताऊं? Photo system 1 से कहाँ गया? बताओ जल्दी बताओ. Yes, I need the answer. FRS. Very good. Photo system 1 से कहाँ गया? FRS में. ठीक है. FRS से कहाँ जाएगा? FRS से जाएगा FD. ठीक है. FD कहाँ पे आया? FD आ गया थैलेकॉइड के मेम्ब्रेन के पास. ठीक है. FD कहाँ आया? थैलेकॉइड के मेम्ब्रेन के पास. Oh, sorry, why I started speaking in English, Hindi. This is an English batch. Sorry. Thylakoid membrane. And here what happened? NADP synthetase enzyme was there. So what happened? NADP positive plus 2H positive is getting converted into Na. One second, what happened? NADP H positive plus H positive. Okay, this is how it's converted. I have written it. Clear? This was there. Now let's see that from where this electron was coming. This electron was coming from the photolysis of water, right? Photolysis of water. And where the photolysis of water was happening, sir? It was happening in the lumen. Okay. Now, the major purpose was to increase the concentration of H positive ions in the lumen, right? So here concentration of H positive ions was increased, and when the concentration of H positive ions is increased, the F0 F1 complex. What happened to my pen now? This time is very fishy, I think so. Yeah, it's working. So now what happened? Through the F1 F0 particles, this H positive were released, and here we had an enzyme which was ATP synthetase. Yes or no? Yeah, ATP synthetase, and this enzyme converted ADP plus inorganic phosphate into ATP. See, Z scheme is clear. Z scheme is clear. We could have completed it yesterday only, but yeah, it was not a good time yesterday, so we completed it today. Any doubts anywhere? Shri Shai, Shivesh, let me know if you have any doubts. Is it clear? How much percentage you are able to understand? 100, 90, 50, 20? How much percentage you are able to understand this concept? Tell me in the comment section. Tell me in the comment section. And do you know what? We have a great surprise for you. Maybe it will come today or maybe tomorrow. I don't know. But a very good surprise for you is coming tomorrow or maybe today. Right? The team is going to... Basically, I'll give you a hint. You know your physics teacher, right? On this channel, Uma Shankar sir. 95% clear. Okay. The 5% will be clear when you're going to revise it. Okay. But in case you have any doubts during the revision also, you have to ask me. Okay. Okay. So, Uma Shankar sir and me, that is Harsh sir, is bringing a very tough challenge for you all. So, that challenge video will be out very soon. And let's see who is going to take that challenge with us. Okay. Now, not more, I'm not gonna say anything more about that video because it's a surprise for you all. Okay, everyone? Great. So, tomorrow, don't forget, at 11 o'clock, tomorrow at 11 o'clock, I am going to take a live class in which we will be solving some genetics questions. The questions will be kind of mixed. It can be from PYQs, okay? It can be from other standard books. So, we will try to build a practice so that whatever the question comes from multiple allelism, or whatever the question comes from codominance, dihybrid or monohybrid cross, we will be able to solve it. Okay. So, see you everyone tomorrow at 11 a.m. I don't think I need to know how the ATP is synthesized in the, yeah, don't worry, we don't need to know that. Yeah, just skip that. Okay, whatever you need, I'm gonna tell you. Clear? Okay, there is lots and lots of things left in the Z scheme. I have just given you the basics, right? Slowly, step by step, we are going to know all the things which is happening here. Okay, but the things which are not present, I'm not going to explain you, because we have to become a doctor, not botanist or zoologist, right? Clear? Okay, great everyone. Physics timing, physics timing is at 3 o'clock. Uma Shankar takes physics at 3 p.m. One short video he has made, I think so, for unit and dimension. You can watch that. And daily he is taking class at 3 o'clock. Okay? Okay, and don't forget to share this video to your friends. Till then, bye bye everyone. See you in the next class. Bye Shri Shai. Bye Shivesh. Bye everyone.